Hello and welcome to the joint colloquium series of the Minnesota Center for Philosophy of Science and the History of Science, Technology, and Medicine program here at the University of Minnesota. I'm Alan Love, the director of the Minnesota Center for Philosophy of Science, and it is my great pleasure to welcome you to this colloquium presentation. Ann-Sophie Barwich is our speaker. She is coming to us from Indiana University, Bloomington, where she's in the Department of History and Philosophy of Science and Medicine, where she specializes on empirical philosophy related to perception, neuroscience, cognitive, and generally sensory science. She is also a member of the cognitive science program at Indiana University. I would be remiss not to highlight, for those of you who are not aware, that she published a book last year, Smellosophy: What the Nose Tells the Mind, from Harvard University Press, which has been getting a lot of attention in a number of outlets. And also she has published a number of articles in diverse outlets, both scholarly and public, communicating the results of her research on olfaction and how it is conceptualized, in fact, I think has been invited to do a whole lot of podcasts as a consequence of the book over the past year. So without further ado, let me turn things over to Ann to talk to us about minding the nose more than meets the AI. Ann, please.
Well, thank you very, very much for the lovely introduction. Let me do the screen share so that everything should work out. So I hope everybody can see the screen. I want to thank you very, very much for inviting me today and giving me the opportunity to talk even more about smell, as many people who have heard me talk about this before. I can talk about this all day, but it's also quite nice for me because I recently finished the autobiography of Paul Feyerabend. And of course, what is mentioned in Feyerabend's biography is also the Minnesota Center. So there's a nice way to connect my otherwise pandemic life and the escape via reading now in a virtual setting back to the community. And one thing I should say perhaps first while we're in the introductory phase is that today is also an interesting day because it's John Bickel's birthday. And there was the biological interest group earlier today, meeting today, talking about a paper of mine on reductionism, which builds on John Bickel's work. So for those of you who actually know John, do feel free to send him some well wishes. It's his birthday today. Now, as Alan nicely said, I published recently a book, and I should, why I'm doing this shameless plug is also to say that if you're interested in the scientific details and some of the issues I'm talking about today, I actually mention in the book in chapter 6. Do feel free to look at this, because one of the things what many people underestimate with the sense of smell is what kind of a recent endeavor it is when we talk about the science of smell. And some of that I will mention today, to what extent there are still some open questions and the changes of the field. Before I start with jumping into the scientific details and the philosophical argument, I would like to start with something that's quite dear to me, namely a note on the philosophical approach that underlies this talk and that underlies this program. And that's what I call the so-called the Churchland program. And the Churchlands are more known for their eliminative materialism and their theories about consciousness connected to materialism. But there's also something I find even more radical about their work, and something which I do hope that philosophers of science in particular are going to pick up more strongly. And that is actually their rather radical idea to rethink philosophy through advances in science. For a long, long time, if we think especially about theories of mind, these ideas came from philosophy, and then there were arguments to find scientific studies, scientific development supporting or contradicting with philosophical ideas. They turned this upside down. They said, well, we experienced a revolution in the 20th century about how we study the brain, what we know about the brain. And that should have an effect on our philosophical conceptualizations about mental phenomena, cognitive functions, and so on. So perhaps it's time to, instead of trying to find empirical evidence for or against our philosophical theories, to derive new philosophical theories based on these ongoing empirical developments. So the Churchman program, as I see it, is to actually think through the empirical material to derive new philosophical perspectives. Also, philosophical perspectives with experimentally testable applications. And that's something I thought is something I always wanted to do, and where the sense of smell offers a wonderful opportunity, because most of the open questions in smell are not just simply philosophical, but also experimental. It's a rare opportunity for philosophers to actively engage with experimental developments. Now, for today, for the roadmap, what I'm going to do is I actually start with a philosophical, not the philosophical argument, but the scientific challenge, meaning what's actually behind the idea of using machine learning in olfaction. That's a trend that's come up recently. And I'm going to look at the ways, why have people been doing that? What's the problem behind it? And also a little bit of a historical background of the field to show what this looks like a reasonable enough idea, before I then actually jump into the philosophical part of it. And it's not a philosophical part in terms of arguing against other philosophers, but to do a conceptual analysis in this particular area. And here the idea is to look at what's the reasoning behind the strategies, and then what's the problem with the current approach. Basically, to look at the conceptual foundations of the field and to provide a theoretical philosophical alternative to current modeling strategies. And that I will back up with some experimental, really recent experimental data on the biology of the olfactory system. Why biology matters, and to what extent the biology actually invites us and even forces us to rethink our modeling assumptions and the theories of olfaction, both with respect to how we model olfaction with machine learning, or perhaps even why this is a limited approach, and also the understanding of perception that stands behind it, and what kind of broader theories or broader consequences we can derive from that. Now let's jump directly actually into the scientific problem, which is actually simply defined as we still don't have a stimulus response model in olfaction. We have one in vision, we have one in audition, we don't have one in smell. And the reason for that is not that smell is subjective, but rather there's a lot of open empirical questions to be addressed here. And the issue here is, how do we actually link the physical stimulus space to the perceptual space? The problem is, well, right now we cannot reduce the odor to the molecular structure of odorants. So odorants is the technical term for the stimulus. And the reason for that is that structure-odor relations are extremely irregular. And I'm giving you an example here. As you can see, these are 4 musk molecules. Now, musk is one of the most well-studied and popular ingredients in perfumery. And as you can see here, these have 4 completely different structural configurations. And these are just 4 examples. So there is no commonality, because as you can see, you've got ring structures, you've got chain structures, you've got nitrogen in musk selene, but not in others. Plus, some of these molecules were synthesized rather recently. So galaxolide, which you see on the top left, for instance, is an invention, so to speak, of the 21st century, because one of the most intriguing things about the sense of smell is that the sense of smell is the only sense for which we can artificially create an entirely new stimulus, meaning molecules that have never existed on Earth. And moreover, they don't even need to smell like anything known in nature. And that leads to even calculations that some researchers recently proposed that the human sense of smell, which has been derided quite a lot of being irrelevant, etc., and in decline, which, by the way, is not true. Nothing could be further from the truth. But the humans can detect and distinguish up to, and maybe even a little bit more than, 1 trillion stimuli. Now, that number has been a little bit in debate when it comes to the precise number. So the official number is possibly shit tons. But it's not just single molecules. So far, I've been just talking about structure-odor relations of individual odorants. But in reality, we seldom smell single molecules. We smell hundreds of them. Coffee, for instance, contains about 800 different molecules, none of which, by the way, smells of coffee. It seems to be kind of an Aristotelian principle that the whole is more than the sum of its parts. Now, what is interesting here is that these mixtures, these kind of aroma mixtures, behave unpredictably. So in this case, for instance, you've got 3% of coffee, by the way, is indole. And indole on its own as a single molecule smells like shit, literally. It has a strong fecal smell, but you don't, of course, smell it in coffee. It kind of contributes to the aroma, but it is not individuatable. So there's no marker. There's like some people are very sensitive to it, so they can detect it, but usually we don't. And that's with the case with many other mixtures. One of them, one good example, one of my favorite examples is one of the most popular perfumes of the late 20th century, Eternity by Calvin Klein. And Eternity contains a lot of this molecule, which is indole. So Eternity literally smells of shit. But what I'm trying to tell you here is not just simply anecdotes, but that we have two problems here. One thing is the relation between single odorants and their perceptual quality, and then how complex mixtures actually behave. And this is why we're still having open questions. And for a long time, was thought, well, this is just because the odor chemistry is extremely complex. An individual odorant has about 5,000 different molecular parameters involved in ligand binding with the receptors. So people thought if only we had better data analysis tools, we might finally crack the code in the nose. And that's the promise of machine learning that people have picked up more recently, where they thought, well, if only we kind of have a good database of these different features, we might be able finally to solve that. And that is really recent. And just to give you an idea of the different areas involved, you have both general science and really kind of highly prestigious journals such as Science looking at artificial intelligence grows a nose. 2017, you have specialist science, Chemical Senses is one of the kind of key journals in the chemical sciences looking at these developments, where human olfactory research has been picking up more and more strands from research in machine learning. But you also have popular outlets such as Wired looking at these kind of trends and even proposing, well, maybe we kind of should jump on board. And the key word here is Google. If Google gets involved in cracking the code in the nose with machine learning, you bet there's money involved. And we're talking about billions. So when people used to question my research as in terms of why are you working on smell? This is kind of like an obscure little sense. No, not at all. So not only have we noticed throughout the year of COVID last year that the sense of smell is much more important to human cognition and human daily life than we previously thought, but also this is $1 billion industry. So whoever cracks the code in the nose can be sure to get a lot of money. The issue here is really to predict, why does a molecule, for instance, like cis-3-hexanol smell of fresh cut green grass? And can we design new molecules with odors? And we can do it in a targeted way rather than tinkering around in the lab. Can we design it on computer and just synthesize it? That would make the whole production quite simple. And you might wonder, wait, why would we need new molecules? And the reason for that is that everything smells. And especially lots of cosmetic and household products do smell and quite often get a signature scent, ranging from high-end perfumes to detergents and shampoo to scented trash bags. And this is just a short, like an overview of the fact that this is not just simply a $1 billion industry, and this is just the US numbers, but also the projection is that it increases. So right now in the US alone, we're over $25 billion per year. So there's a lot of money involved, and you might still wonder, yeah, but why do we need new molecules if we have so many already? One reason is not just simply to create new molecules with novel odors or things that have some novelty factor, but also if you have, let's say, a range of products with the same smell, especially in perfumery, where you've got a high-end perfume, you've got a body cream, a shampoo, maybe something else like a soap, you cannot use the same odorant for these different, or the same kinds of odorant for these different products, because odorants in different chemical environments will behave and smell differently. So there's a lot of tinkering and development involved, and that costs a lot of money, and the hope is that machine learning might shorten that process. The problem with that is that underlying this whole development is kind of the common trope of from molecules to perception. What people try to get together are two kinds of data sets. On the one hand, you've got the ligands with several thousand, like 5,000 molecular parameters involved in binding. On the other hand, you've got all the sensory descriptors. Some of you might already say, wait a second, but sensory descriptors, words are not the perceptions. Precisely. So there's a lot of processing in between molecules and words that is not addressed here, but this is really the two data sets mainly used. So the theoretical point of view is really, from a theoretical point of view, what's the problem is we are excluding that in-between bit, and that matters quite a lot. So the problem, as I see it here, is the underlying notion of perception. So our brain is often kind of modeled as extrapolating information via some kind of efficient feature extraction process. And that's at least how lots of these machine learning models have been construed, that you've got an input such as cis-3-hexanol. You do some kind of feature extraction where you look at the molecular parameters. You compare it with other odorants that have a similar enough smell and see like, okay, what molecular features might be similar? And then do all kind of lead to the same kind of output. Does it have a green smell or does it not have a green smell or any kind of other smell involved here? This is kind of something quite often built on a kind of a simplified model of the visual system. And the question, of course, is that really how olfaction works and how we should model it? This is kind of a big jump. So the idea is really that the sensors are there to filter out general information from contingent scenarios. So you see that, for instance, in the computational model of David Marr, when he talked about vision, you also have this in more ecological-minded models such as James Gibson. The question I find is, well, what are the significant bits and how does the sensory system represent those? And these questions, unfortunately, are currently considered irrelevant for machine learning approaches to smell. But they're not, and this is what I'm trying to show you. You might say, wait, you might be a little bit too unfair and uncharitable to machine learning people working on smell. No, I'm actually not. I can show you that this is precisely what they say. So this is an excerpt from the Google AI blog. So the people with the money who do this and who I find throw the money a little bit out of the window, it's based on analogous advances in deep learning for sight and sound. It should be possible to directly predict the end sensory result of an input molecule, even without knowing the intricate details of all the systems involved. That is quite a big jump. And that basically assumes that we don't need biology to rethink what we're modeling. And I'm going to show you why this is really the wrong way to go and to understand the system. That being said, these are smart people. So what's the reasoning behind that? Well, the strategy actually made historical sense. It made historical sense because the 20th century was the time where people modeled olfaction in terms of its chemical basis. But the reason was there was nothing else to experiment on. The biology was hardly known, and these molecules were the only thing you could properly experiment on, twist and tweak and study. The biology at that time was more hypothetical. It was mainly built on theoretical models applied to olfaction to inform the chemistry. And one of them was, for instance, Linus Pauling, who in 1946 suggested the lock and key mechanism as applicable to smell. Well, obviously, we've come a long time, like a long way since lock and key. It's a bit more complicated today. However, back then people used this to say, well, maybe we should look at kind of structural similarities that are somehow involved in ligand binding that could tell why certain molecules have the smell they do. So one of the pioneers here was actually John Moore, who coined the stereochemical theory of odor, where people looked at molecules with the same shape should have a similar enough odor. That was successful for a while, or successful enough, and people started to look at what's called structure-odor relations. So to find rule-based applications to say if a molecule has these kind of features, it should have that smell. And one of the first successful rules was in 1971 by Günther Olof, a German chemist, who coined the triaxial rule for ambergris. This is a rule that says, well, if you want an ambergris molecule, you ought to have a bicyclical compound with certain atom groups in specific positions. That quickly turned out not to be the case in terms of that there are some molecules that scrupulously rebut this kind of rule. One of them is carinol, for instance. And this is the fate that befell many of these structure-odor relations. And just to give you an idea of how much that actually matters for a fragrance chemist, this is a page from the notebook of a perfumer. It was in the 1990s, so of course, this work would have been a bit easier now with kind of more high-tech computational methods. But his job was simple: design a sandalwood molecule, because there was back in the time lots of money to be made from sandalwood odorants. And how that happens is you've got a parent molecule, and then you twist and tweak it a little bit in terms of structure similarities to get something out of that that hopefully smells like sandalwood. This is the entire year, the production of entire year of a fragrance chemist. And only one of these molecules smells of sandalwood. It's not obvious which one. So that just to give you an idea of how difficult it is to find a good relation to say like, okay, this structure might cause that kind of odorant, and why people said this might not work. And then the big revolution happened. And this revolution, unfortunately, hasn't been taken into account properly today. Namely, things have changed in biology, because in 1991, Linda Buck and Richard Axel discovered the receptor genes, namely the olfactory receptor genes that identified odor receptors as part of the largest multi-gene family in the mammalian genome, G protein coupled receptors. And they're involved in a variety of important functions, such as ranging from vision to the regulation of immune responses. The adrenergic receptors are GPCRs. So this is an important family, and that catapulted olfaction from being this kind of obscure little system a couple of people were working on into mainstream, into hardcore mainstream neurobiology and neuroscience. And not only are GPCRs the largest family in mammalian genomes, so GPCRs are up to 10% of the genomes, but the odorant receptors, the odor receptors themselves, are even individually, taking aside the largest multi-gene family. An exception in the mammalian genome are dolphins, but otherwise it's quite astounding how many members this family has, such as 400 members in humans. Just by comparison, you've got 3 cones in vision, 1,000 mice, 2,000 even in elephants, which is pretty cool. Point being that people thought, oh, finally, we've got the receptors. We can finally model the input-output relation from the air to the brain. We've got the key that unlocks this kind of relation. And then nothing happened. All these kind of hopes that we can march through the system and solve these kind of regularities by having the receptors didn't work out. 1990s, nothing. 2000s, nothing. We're now in 2021, we still don't have a structure-odor relation. So nothing happened, and the question's why? What's the case here? What's the difficulty? And this is not just simply an assessment by me, but you also had fragrance chemists who noticed that. And one of them is Charles Sell, who's now retired, but was one of the key people working in fragrance chemistry, who even wrote a paper called On the Unpredictability of Odor, where he explicitly said there's too much structure odor irregularity. We're not getting there. And then people picked this up, because people were, well, maybe we got the wrong tools. Now that we've got machine learning, we might finally go where our predecessors couldn't have gone before. And I'm going to show or argue that this is actually problematic, because the idea of structure-odor relations doesn't apply to modern olfaction anymore because of biology. So there are 3 problems of these models I want to walk you through next. First of all, the problem of the data and the data sets we're using. Second, the presumed link between these data sets. And third, the task, the olfactory system, the kind of problem we're trying to solve. All 3 actually point towards that we should abandon the idea of neat structure-odor relations. So let's start with number 1, the problem of the data. And there were increasingly studies. It started especially in 2011 with a paper by Alexei Koulakov and Dima Rinberg, which applied machine learning to a data set of, okay, we've got these molecules, we've got these descriptors in search of the structure of human olfactory space, linking physical stimulus space to perceptual space. So why does heptyl acetate, for instance, smell of pear? Well, you can already see what's the problem here is we're excluding the biology. We're excluding the psychology, and the question is like, why should that matter? Well, first of all, the data is limited, and you will see with many of these studies, so this is not the only study, that they're all referring to the same kind of data set being used, and that is the Atlas of Odor Character Profiles by Andrew Dravnieks, which was published in 1985, notably way before the receptor discovery, which is, by the way, not a database. It is a book that lists basically 146 verbal descriptors of odor quality for odorants. And these verbal descriptors were collected by a single individual, namely Andrew Dravnieks, which is a great thing. I mean, he contributed quite a lot, but it's basically the perceptual space of an individual person. And he applied that. I mean, he tested that with a couple of other subjects. But first of all, these descriptors are outdated. They are built on the baby boomers generation. They also are limited in terms of what descriptors are being used and also what odorants are being used. And it was not intended for structure-odor relations, but it was supposed to be a commercial thing in terms of how do we help people talk about odorants in commercial context, in consumer production, consumer negotiations. And just to give you an idea, for instance, there are lots of molecules and descriptors that are culturally very specific. If you look, for instance, at the notion of bakery as a descriptor for some molecules, in the American context, bakery refers to vanilla. You would not get that response from somebody who, like me, is from Germany. No. When you think of fresh bread, you have a much more savory odor connected to bakery than vanilla. The second part is like, well, okay, fair enough. You've got the wrong data, but that's not necessarily an issue of machine learning. What if we had better data? What if we had real empirical data, like a proper psychophysical study we could investigate? Well, that has been done. And very recently, the study was published in 2017 and was a huge project. And so far, the best study available on that topic, where they used new and an extensive set of psychophysical data. For olfaction, at least, it's a big one. And they also are quite interesting because it was one of those crowdsourcing efforts where you've got some kind of participants from lots of people in and outside of olfaction addressing this challenge. And as you can see, they're quite bold with their title, namely predicting human olfactory perception from chemical features of odor molecules. They didn't predict anything. But let me explain that kind of strong statement. So first of all, what was the setup? So the setup was that they got two sets. They've got the set of molecular features via the software eDragon, and the psychophysical data set, where they had the descriptors on how people assigned descriptors to these molecules. And the task was, well, find an algorithm. Then they got a second psychophysical set to refine the algorithm. And then what are the structure-odor relations you've got from that? And the data was like you had the eDragon data set for the molecular features, and then you had a study by Keller and Vossel from 2016, which for olfaction is a big data set. And I highlight that because people who work in other fields might say that's not big enough data. But olfaction is hopelessly underfunded, especially psychological studies. So this is where it was the biggest one so far. It had 49 subjects, 476 molecules, and 19 descriptors from Dravnieks. What's the problem with that? Well, first of all, the success rate was 0.3, which is not really successful. First of all, one of the most successful predictions was that most of these molecules used also in perfumery smelled floral. What a surprise. And that molecules with sulfur in them smelled sulfurous. So there was a lot of confirmation of things already being known, but there was no prediction in terms of that would help you to predict new molecules, or that would predict the smell of new molecules. And one of that is the absence actually of a good psychophysical theory, because you had a variety of descriptors, 19 odor descriptors, but you had also the question about how could we relate this to pleasantness and intensity, which are different parameters, different dimensions of olfaction as a function, because you have hedonics, you've got as hedonics in terms of do you like it or do you not like it? Is it pleasant or unpleasant? You've got the quality and you've got intensity, but these are different dimensions of olfactory functions, and they're also not processed the same in the brain. So the question is, can you really predict all of that with molecular structure? You also don't have a good categorization of broader terms and smaller terms. So some of them are fairly broad, such as chemical, which is a descriptor just used for, do you detect anything, even if you don't quite know what it is, to something very specific, such as garlic, and something more broader, such as, for instance, warm in fruit. So there is no good psychophysical theory that would elaborate on the categorization behind these notions. And I actually asked Andreas Keller, the lead author of that study, why these 19 descriptors? His answer was, I like them the most. So not everything is necessarily methodologically that elaborated in these kind of studies. And the problem is that you remain with an unknown olfactory space, so that this data alone will not give you a good structure-odor relation, or at least model for it. And one of the critics who put that publicly is Avery Gilbert, who's a sensory scientist, who, if you're interested in the topic of olfaction, his blogs are a hoot. First Nerve, who wrote actually criticism. Can we predict a molecule's smell from its physical characteristics? He said, nah, there are lots of problems with this study. One is, if one wants to predict what molecules might smell of, let's say, sandalwood or citrus, not mentioned here, one would have to retest all 476 molecules on another 49 sensory panelists using the new list of descriptors, then rerun the computer models on the new data sets. So this is actually a very limited way of doing things. But the more important part is that we're still trying to connect words to these kind of data sets. And words are problematic because they operate on different levels of cognitive characterization. So we still assume that the word pins down the perception. And I'll give you in a moment a couple of examples why that is not the case. Namely, we're still assuming that smells are somehow down to stimulus chemistry. We just need to find a good way why this kind of set of chemicals elicits that kind of olfactory quality. Well, first of all, we need to get rid of the idea that the semantic object we're having or describing with these kind of words is the same as the causal object. Because one nice thing with smell is that you can have actually a lot of different smells being elicited by various kinds of compounds. So you might have something like lobster that can come from a real lobster. It might also come from a perfume that an evil perfumer has designed to fool you into thinking that is lobster, and that perfume will not need to have the same odorants being emitted as those from a lobster. By the way, that's a real perfume. So this is a series of conceptual perfumes. They also include fresh laundry or even funeral home. I kid you not about the funeral home. The next thing is that odorants are actually semantically ambiguous when it comes to their quality. And this is one of my favorite studies done by the psychologist Rachel Hertz in 2001, who basically gave people two vials. Looked identical, so same liquid, same color, same bottle, but the only difference was the label was different, meaning one was called Parmesan, the other vomit. And participants in her study were convinced, well, these two bottles have a different smell. There's something different in there. They also had different hedonic reactions. So most of the people, of course, preferred the Parmesan over the vomit. What they were later told and didn't believe at first was, this was the same mixture. This was a mix of isovaleric and butyric acid. So same mixture, different smells. And she did that with a couple of other molecules as well. So she had patchouli, which was musty basement, and something which I'm currently blanking on. Then she had mint, which was, oh yeah, Christmas and wonderful happy family, versus dentist office. And people, again, this is different. It's often seen like, oh, well, this just shows like, well, you can twist people's perception around and it's unreliable. No, the point is that the chemical stimulus is promiscuous. It's promiscuous in the sense that you can have the same odor molecules occurring in different contexts, in different molecular clouds, having a different behavioral relevance. So it's not a matter of being fooled. It's a matter of your system has to react flexibly to contextual information. And that means you have to look at how the sensory system makes sense of these, literally makes sense of these molecules. And that means it's not just simply the stimulus, but it's a computational feature. It's a feature created by the system. And the best example for that is the case of olfactory white, which was discovered in 2011 by the lab of Noam Sobel at the Weizmann Institute, who realized that, well, if you have 30 molecules with any kind of set of molecules, the only difference is that they don't have overlapping causal features. So specific functional groups they shouldn't share and all these kind of things. And if you have 30 molecules with non-overlapping causal features, at the same intensity, it smells of something you don't know from nature. And he called this quality Lorax. Then he had 60 molecules, same principle, non-overlapping chemical features, same intent, like it's equal intensity, and it smelled the same. Then he arbitrarily divided this mixture of 60 plus molecules into two, and the two mixtures, which were arbitrarily divided, both smelled of Lorax. He did that with 90 molecules, same principle, so on. So what this shows is that odor quality is not just simply stimulus chemistry. It's how the biology makes sense of the chemistry, meaning kind of a computational feature, computational in the kind of neural sense and a bit loosely used here to understand what's actually being produced with the chemistry. And this is precisely where I come to the last thing. What we need to model is the sensory system that gives rise to these different qualities. And of course, that elicits a very, very simple question, namely, is there actually sufficient reason to think that knowledge of receptors and their behavior, and of course, in extension, also the rest of the sensory system, affects and changes our understanding of odor coding? The short answer is yes. The long answer is actually also yes. And I'm going to walk you through how that also affects our models of machine learning. So, but before I can do that, I should say a couple of things about how we actually process olfaction or the olfactory stimulus, since it's one of those underexplored sensory systems, and not many people actually know much about the details of olfaction. Sure, it's something with chemicals, but what does that mean? Well, in comparison with vision is better known in terms of it has a low-dimensional stimulus. Low-dimensional meaning you've got one key physical parameter on which you build or base your perceptual models. In this case, it's the visible spectrum of wavelengths. And you've got three current receptors, typically three, in humans that basically detect different chunks on that kind of visual spectrum on which you can build then your perceptual theory. Similar with audition in the sense that it also has a low-dimensional stimulus of frequency, which you can map actually quite nicely in the auditory cortex. What about smell? If you have something like, let's say, rose smell, you actually have usually when it comes to natural roses, 400 different compounds, sometimes more, but hundreds of compounds being involved in rose smell. Even synthetics are often a dozen molecules that you need together to elicit rose smell. And as I mentioned, each odorant has 5,000 parameters. So this is like big data for your brain to process. So how does the brain actually make sense of all these kind of different clouds and chemical features? Well, the really honest answer is we don't quite know yet. There has been a mass of new studies over the last decades. And since the receptor discovery, it's like precisely 30 years, by the way. Next month, on like exactly 1 month from now, we actually have the 30-year anniversary of Buck and Axel's paper. So since then, a lot of things have been done. Why we don't know yet what that means is because we still thought we can just apply concepts from vision. And what turns out to be the case is that, well, smell works a little bit different than vision. So it requires us now to rethink and re-understand the data we actually got from that. And it looks deceptively simple, but the devil is always in the details, because what happens when you smell is you've got odor and molecules that travel up your nasal cavity and interact with the sensory neurons, with the receptors that are situated in the sensory neurons in the nasal epithelium. And what's happening here is basically that you have each sensory neuron expresses only one receptor type. And all the sensory neuron expressing a specific receptor type converge in particular areas in the olfactory bulb, which is situated at the inferior frontal lobe of your brain. And these they converge in one place, and these places are called glomeruli. So they're kind of like little neural structures, so to speak. And it looks almost as if you can get a neural fingerprint in your brain of these molecules. And then you go to the cortex, specifically the piriform cortex, although I should say this is actually the insect part, but insects and mammals have this kind of shallow three-level pathway. But in the piriform cortex, it just goes bonkers. It goes in all kinds of different directions. So unlike vision, the somatosensory system, unlike audition, you actually don't have a topographic organization. It just goes in all kinds of directions. You've got almost like a disco brain. It's very mosaic. It's beautiful. Point here being is there are lots of open questions, and we should look at how this system actually decodes that kind of information. And this is why we should start actually with receptor biology rather than just looking at the kind of broader theoretical model. Because what happened in the last 10 to 15 years is that people just look at the central nervous system and look at the higher up processing as going like, well, you know, the receptors, there's just some kind of decoding, but we follow the signal and we figure out what it is. Well, it turns out that this didn't lead to any good models of the olfactory stimulus or stimulus representation in the brain. And so people start to go back to the receptors because something is happening here. And the key issue is that for a long, long time, we thought that the stimulus chemistry is somewhat coextensive with odor coding, meaning, well, clearly the receptors just detect the chemical features. We can also model with the chemicals, right? Well, that's actually an empirical question. And it turns out that empirical question gives us a negative answer. Because not only do we not have a stimulus odor model, but we can't even tell from the chemistry alone whether a molecule has any or some form of, or is odorous at all. So this is a very good quote that actually exemplifies that. It is nearly impossible to predict whether a given molecule will be odorous and what its odor quality might be from the chemical structure alone. Although all odor molecules are typically organic compounds of low molecular weight, they may be aliphatic or aromatic, may be saturated or unsaturated, and may have any of several polar functional groups. However, there are many molecules that conform to those characteristics, which are nonetheless odorless to humans and other animals. So something is missing here in our models. And to explain that, and why that is so important is a good way to do that is to compare it with a visual stimulus again. In vision, as I mentioned, we've got a low dimensional stimulus where, let's say, you've got red light and you can pin it down to a particular wavelength spectrum. We say, okay, this is red, this is the range of red, this is what the red cones basically detect. And that allows you to derive more kind of well-defined models of other colors, even colors that don't have a wavelength. So one of those is pink, for instance. And pink doesn't have a wavelength associated with it, but it's a computational feature of your brain where you've got white light minus green light results in pink. The problem with that is that in olfaction, we don't have this kind of neat additive calculation because of the complexity, because of these kind of multidimensional, because of the high dimensional stimulus. And a good example for that is aldehydes. Again, one of the kind of key ingredients of modern perfumery. And for those who like Chanel No. 5, it's actually consists almost entirely just out of a bunch of aldehydes. One of the first molecules, perfumes designed with new synthetic compounds. And here you've got basically, let's say, you've got an 8 carbon atom aldehyde, which has a fatty smell. If you've got 10 carbon atoms, you've got a citrusy smell, and the longer the chains get, the more floral it gets. But you don't have a neat kind of feature addition model here. It's not that you can make something like, oh, well, you've got 8 carbon atoms and you add another carbon atom, therefore cherry. This is just not how it works. So you don't have this kind of same combinatorial in the kind of linear additive sense. And the question, of course, why not? And this is the answer lies actually in the receptors and how the receptors respond to the stimulus. In vision, you've got a fairly uniform tuning range of like, okay, you've got different chunks of the visible spectrum being detected by specific cones that then allow you to combine it up. In smell, first of all, you have combinatorial coding. Meaning you've got one odorant can be detected via different features by a variety of different receptors. And one receptor can detect different features of different odorants. That explains why you have this explosion of different combinatorial possibilities in smell. That explains why you can perceive so many different kinds of molecules, and also why you don't have to grow new receptors the moment you smell some new perfume. I mean, perfumery would be a fairly problematic business if you have to wait for people to kind of develop some new receptors, so to speak. But you smell it instantly, even though it's like synthesized in the lab 5 minutes ago. The next thing is it's not just combinatorial, but you have wide ranges when it comes to the tuning of the receptors. Some receptors detect just a few features. Other receptors are fairly unselective with the different features they can detect. So you don't have a uniform odor chemistry in the receptors, which is unlike vision, where it's got a fairly uniform distribution. In smell, it's like all over the place. Plus, the olfactory system is one of the most genetically diverse systems, meaning you and I, we don't share the same set of receptors expressed in our nose. So we literally perceive the world differently because we're genetically diverse, we don't have the same receptor repertoire, and even you yourself, if you move continents, will over time actually change your receptor expression pattern in your nose, because adult neurogenesis, another nice feature of the olfactory system. You might want to, okay, but what has this to do with odor quality? Quite a lot. So one of the coolest things is that receptor genetics influences how you perceive a molecule, the quality you get from a specific structure. 2 examples here. One is cilantro. You know, when you've got these people, or coriander, where you've got these people over dinner and you've got always one person who's like, yeah, I don't like cilantro. It's kind of pungent and soapy and I don't like it. These are not picky eaters. They're mutants. They have a mutation near one of their olfactory receptor genes, leading to a difference in the sensitivity in responding to these odorants. So you might, I mean, you've got, this is just one example, but another one is androstenone, which is a pig pheromone, which humans can also smell. Most humans, there's some people who can't smell it, and there's some people who can, and those people who can, turns out, perceive it completely differently. So you've got some people who think, it's very unpleasant because it smells urinous. Other people find it very unpleasant because it smells like body odor. Then you've got people say, oh, I like that smell. You know, it has kind of a woody smell. And then you've got people who find it more kind of floral or vanilla-like. So not only do you have different hedonic evaluations in terms of liking, disliking, but you also have different categories of perceptual quality. And the reason for that, it was recently shown, has to do with genetic diversity, informing or instructing receptor sensitivity, leading to different qualitative features. So in other way, the whole issue of just mapping these kind of 2 data sets of ligands and verbal descriptors is excluding the key causal element, namely the receptors, who do not perceive chemistry like an analytic chemist would necessarily also model the molecules, not just because of different sensitivities, but even in terms of what features are being detected. So there would be, just to say, the receptors detect the same or respond to the same features in the same way a chemist would model an odorant in terms of what are the relevant chemical features in terms of causally responsible. It's an empirical question whether that's the same. And 2 recent studies, 2016, 2018, so these are really recent studies, have shown that actually, if you look at, and this is a study on ketones, if you look at how chemists, for instance, would model the similarity between different ketones, and then you look at how the receptors classify them in terms of what they like to respond a bit more and consider to be more similar, you get a completely different similarity tree. So biology clearly does not care what an analytic chemist does. And you might say, well, what does that actually mean in detail? Well, one thing is a chemist would model ketones in terms of, well, first you separate them by the ring size of the molecule. Then you look at the composition of the ring. You look at subfamilies of this molecule. Let's say have a nitrogen, an oxygen, a sulfur atom, and so on. What do the receptors do? They don't even care about ring size. Ring size doesn't matter at all. Neither, by the way, does cycle composition. What's most important for the receptors to respond to something and accept it is the polar surface area, which is the electric charge in the 3-dimensionality of the cycle. Does the neuron accept actually the odorant or not? So you have a feature that, by the way, wasn't even coming up, wasn't even predicted in that big 2017 machine learning study. So if you think that machine learning doesn't care about all these kind of theoretical things, and it's like, well, it's a good way to circumvent our models. But apparently not. There are some features that machine learning, for instance, didn't detect despite having all the data. So to sum up, the short answer is that, first of all, our machine learning models have been largely built on vision, and this is already quite theory-laden. So olfaction is not vision. There is no homogeneous feature extraction that would lead to an additive stimulus model, or even kind of a fairly computational, straightforward structure-odor relation. And if you're interested in more examples and a more elaborate even discussion of that, I've got not just the book, but also a critique of olfactory objects, which is a paper that was published fairly recently as well. That just as an emphasis on why we should stop black box, why we should not black box the biology of the system. Because first of all, what constitutes chemical similarity differs between the principles of organic chemistry and receptor chemistry, so to speak. And in fact, when we think about the human olfactory system, you have 400 different types or different kinds of receptor chemistry. So it's important to kind of take this into account. And since I'm noticing I'm getting slowly towards the end of the talk, let me at least sum up a couple of smaller things of the long answer, which I will have to keep shorter. But if you're interested, there is more to get out of there, because this was the short answer. The long answer, like, well, does it even matter in terms of other processes that might help us understand olfaction? Yes, in fact, hell yes. Because as I mentioned before, odor chemistry, odors, like the sense of smell, is not just about detecting single odorants, but mixtures. And this is one of the most interesting things, because if you have something like, let's say, the smell of potato chips, and this was a study done by a colleague of mine, who said, well, you can actually create a complex image, a perceptual image like potato chips, with key odorants that have in and of themselves nothing to do with potato chips. So you've got, for instance, rotten cabbage, potato, toast. The point is, you don't create that image by just having a set of molecules, but they have to come together in a specific ratio. So clearly something is going on in terms of how the system combines features together. And that cannot be simply explained by the combinatorial system. Why not? Because of the ambiguity. So in a combinatorial mixture, for instance, you've got some kind of overlap between different molecules that activate similar enough receptors. And that can lead to an interesting combination of mixtures where you might have two mixtures with different components eliciting the same receptor repertoire, if you just go by the principle of combinatorial coding. And this is kind of just a hypothetical example. Let's say you've got odorant mix of odorant 1 and 2, which would actually elicit or would overlap in receptor activity with O1 and O3. So you've got an overlap. So how does the brain actually then know what it smells? There is an ambiguity built in the system. How does your brain know what the nose actually smells? And this is one of the things that was recently actually investigated by the lab I'm coming from, by Stuart Feuerstein, where a new technology called Scape actually looks at how does the receptor sheet, how do the different sensory neurons respond to a chemical mixture, which long was impossible. For the reasons basically in terms of it was technically impossible. And now this sheet actually allows you to not just scan intact cell tissue, but also have receptor populations as well as single cell resolution. This is astounding. And that was published in 2020. The instrument itself was devised in 2015. So this is very, very recent. And this is, by the way, the lead author of the study. And they basically looked at over 10,000 receptor cells to see, okay, how does it respond? Turns out there was a big surprise. Not just was there some kind of suppression effects, but also enhancement effects. And that overall, 20 to 30, and sometimes even up to 38% of the cells responded differently to the mixture of three components than in comparison to the individual components. And that was quite surprising. So what they actually found out is that you have not just simply mixture-dependent agonism and inverse agonism, so reduced selectivity, but also the modulation in terms of allosteric effects. And I will have to skip through a couple of things, but just to explain to you why, for instance, that matters is What you're having here is, when I say enhancement, is you have cells responding to a mixture that do not respond to the individual odorants. So you have more cells responding, and sometimes 20%. So this is a significant effect. Why does that matter? Well, it might explain why certain compounds, for instance, have a configural effect, where if you've got a mixture like a perfume, it smells like nothing like the individual compounds, but adopts a quality you wouldn't have predicted from just, let's say, you've got a little bit of a peach molecule and a carrot molecule and what have you. And you might get, let's say, instead of something fruity, you might get something woody or something. So this was one of the kind of key things. And it turns out that the receptor behind it seems to be, at the moment, unique to olfaction in comparison to other receptor systems, to other sensory peripheries. Why? Well, it's a mechanism that's known from other areas in actually medicine, when we think about medicinal chemistry of drug target design, but it's not known at the sensory periphery. So vision doesn't work like that. Taste doesn't work like that. Audition doesn't work like that. So it seems to be unique to smell, namely that if you've got a mixture and you've got a receptor that doesn't respond to odorant 1, but if you throw in odorant 2 in a specific mixture, this second odorant, this second compound binds to the allosteric side, which is a hypothesis binding site that changes the binding site of the receptor. There's a configural change such that suddenly, in combination, the receptor then responds to that odorant. You might wonder, what's the point of that? I mean, what does it explain on this theoretical level? Well, what it explains is how you can resolve the issue of ambiguity in mixtures. You must imagine if you've got a compound of like a mixture of 800 compounds, how does the brain how might it be separate? How might it be able to separate this complex mixture from another complex mixture, given combinatorial coding, given that you've got hundreds of receptor types being potentially activated? So how does the brain solve that? It sparses the signal. By inhibition and by enhancement, it separates one mixture from another by activating, for instance, cells that wouldn't be activated by the other mixture, or it inhibits certain cells otherwise activated by the single compounds. This is a way in which you can explain seemingly higher level perceptual effects on the sensory periphery. And that has been one of the most revolutionary studies in the last time. Broader implications of that is not just simply that you might be able to link biological effects to material practice, perfumery, wine tasting, winemaking, all these kind of things, but also, there it is. It shows why we should rethink machine learning. Why? Because this was kind of a long story. Well, first of all, it shows to what extent you cannot just simply connect the structure of molecules with some kind of psychophysical data set. Because the molecular features you're modeling might not be the ones that the system is detecting. And also the perceptual effects are not directly linkable to the molecular structure. And if you think, well, I'm a little bit unfair to many of these kind of machine learning studies, the problem is that they're getting too much attention. The problem is that this is where the money is currently going and which hinders progress in olfaction. And there's a lot of people who are jumping on that. You've got not just Google, but IBM doing precisely the kind of modeling I've been criticizing, not looking at the biology, black boxing the system, and it harms the field. And it's really kind of the old saying of garbage in, garbage out. Like, you can apply these models, yeah, but you're not even getting good correlations. So you might say, well, okay, so we got the wrong data. What if we just had better data? Maybe at some point we might be able to do the whole machine learning thing in a better way. And I would like to throw something in which is a little bit more provocative. Namely, this is like saying, well, we could be Laplace Demon, and if we had all the data available, then we might get to a proper model. Well, first of all, the data we would need is not just simply accounting for the genetic diversity of all humans, but all possible mutations that might lead to different sensitivities in the receptors in response to odorants, leading to different qualitative perceptions of otherwise the same molecule. And this is, I gotta say, a nice metaphysical picture for hypothetical in principle sometime in the future, if we were omnipotent and had all the knowledge in the world. Yeah, but this is not how we do science, and this is also not how science works. So these kind of in principle theoretical arguments just show to what extent this is actually unfeasible in practice, not just right now, but even within the next 20, 30 years. So you might say, okay, so what? What's the point of that? And this is where I end my talk, because I do think that this kind of story has broader implications. You might say, okay, this might just apply to olfaction because it's this kind of very complex system and we have to think about biology, but I work on a different system. What's the point? Well, first of all, it shows to what extent we really, really need to be very careful about the logic of research questions, which is a term I adopted from my colleague here at IU, Elizabeth Lloyd, with whom I want to actually write this topic up in terms of the light of logical of research questions, like how can we actually analyze different modeling approaches in terms of how it differs in terms of how we ask questions about a system, but also it questions the role of expertise in philosophical studies of so-called case studies. Because this is not just simply a case study. This is a way of applying philosophy from within a specific ongoing experimental inquiry. And that leads me to end with a bit of a provocative question, because I come from a place that was more in terms of pluralism, namely Exeter, Eugenics at Exeter, which is the pluralism and anti-reductionist place. And I seem to currently kind of go a little bit against my own training. But this also shows to what extent certain forms of pluralism, that 1,000 flowers bloom. Well, let these kind of machine learning people just continue with their structure-odor relations. Why not? Maybe they can come out with something. I say no, actually, there might be some forms of model pluralism that can be harmful. Because in this case, we are really burying the lead. We are asking the wrong questions. In fact, we just follow a certain form of model that might have been applicable prior to the receptor discovery. But we need to ask better questions about when a modeling strategy is false, when we have to reduce pluralism. And this is not an argument about pluralism per se, but in order to make pluralism more valuable and practically applicable, we need to rethink of when we need to reduce a certain form of broader plurality. And that it can be harmful, I actually explored in another paper when it comes to structure-odor relations, where there's the public discourse about the so-called vibrational theory of odor, where the case was being made by Miriam Solomon, one of the kind of more popular and well-respected pluralists in the history and philosophy of science, who tried to show that, well, we can have competing models and that might enrich the field. And it turns out actually that this has been so far removed from the practice of olfaction that it actually turned out to be a harmful representation of the field. So this is kind of a provocative endpoint I would just like to drop into discussion potentially. And with that, because I am now way overboard and I would like to hear some of your questions, I would like to also thank the stewards, the Firestein Lab at Columbia, especially the people I worked with, such as Lu Shu, who's been the lead author of the mixture study, as well as Alban Poivet, who's been doing the whole ketone ester study as well. So with that, thank you very much. Thank you for your patience. And I'm very, very much looking forward to your questions.
Thank you, Anne. So now we move into the question portion of the presentation. I already see some hands raised. I just wanted to highlight that at any point you can click on the raise hand function and I will be able to see you in the queue of hands. When I call on you, I will unmute you and then you will have the opportunity to ask the question. If for some reason you would like to ask your question in the chat, you could do that as well, and I will do my best to toggle back and forth between the two. Our first question comes from Ron Okagaki. Ron, you should be able to talk now. Ron, can you unmute?
I'm sorry, I must have hit it accidentally, so it's not a question, but thank you for your talk.
Let's move then to
Thank you.
Miriam Solomon, who is next. Miriam, you should be able to talk now.
Can you hear me?
Now I can.
Now you can hear me. Okay. Yes. So.
Oh, shoot.
Oh, I can't. She's gone.
Miriam, there was a problem. Can you restate your question?
Oh, the last time I met you, you were beginning all this work, and it's really super interesting. I'm delighted with where you are. And I wondered, since I was thinking about the Lukaturin kind of side attempt to understand it, he wanted to understand rather than structure in terms of the vibrational frequency of the molecules. And from your point of view, he's also completely missing the biology.
Yes.
Is that how you would describe it?
Partly because he misses the biology, and partly also because he addresses a problem that was relevant in especially the 1970s. And I mean, Turin, I actually found his model incredibly nifty. It reads beautifully. The problem is that he models biology like a physicist would. Like there's the one feature that rules them all. The issue here is that partly he doesn't take the receptors seriously. He doesn't take biology seriously enough, and he still tries to solve a problem that was a problem because we had the wrong assumption in terms of still trying to figure out what makes the chemistry similar, rather than, well, maybe we got the wrong understanding of how the causality works. So yes.
Thank you. Thank you very much.
Also, thank you.
Okay, thank you, Miriam. Let's move on to our next question from Dan Brooks. Dan, you should be able to talk now.
Okay. Hello, Anne. Thank you for the fantastic talk, and forgive the pun, I have to say it's a phenomenal topic.
Oh my goodness.
My question is based on total ignorance because I am in learning mode and you are the expert on this. So one of the modes of perception, sensory perception you did not mention was taste. And I'm wondering what kinds of work, if any, have been performed in conjunction on olfaction in conjunction with taste. And I'm wondering specifically whether or not, the sort of specialists who enjoy wine, whiskey, or cheese would offer any kind of interesting psychophysical set. Thank you.
I love that question. So the short answer is yes. The longer answer is there's been some fantastic work, and if people are interested in the conjunction of olfaction and taste, the person to go to is Linda Bartoshuk, who's like the pioneer when it comes to taste psychophysics, and she moved more and more into olfaction. And she's one of the people who actually told me, well, one of the most fascinating things that will have applications for, if we think also about how we might be able to change people's preferences with nutrients and obesity crisis, etc., is that you can influence how sweet somebody perceives a certain kind of meal, a certain kind of food products by introducing certain odorants. So you're not increasing the sugar rate. You might just have, let's say you've got a drink and you have like a smell, let's say you've got a mug or something, and you would have some kind of odor coming from, let's say, the rim or something, and it's a specific kind of odor. People might then perceive something as sweeter. Now, sweet is not a smell, sweet is a taste. So there's a lot of cross-modal interaction going on that you can shift people's perception as enhancing or reducing certain things. There are differences between taste and smell. So you do have, for instance, certain forms of agonist and antagonist responses also in taste. However, you don't have the allosteric response when it comes on the molecular level. So long answer short, because I could go on otherwise, is there have been studies. I exclude it because it's already far too complex to put in one book, but there is more to get out of cross-modal studies, and I here recommend not just Linda Bartoshuk, but also Charles Spence at Oxford, who does lots of the cross-modal stuff, and you got the Ig Nobel Prize for the CRISP study and the audition. And there's also Barry Smith in London, who I really recommend, especially when it comes to thinking about wine. He does a lot of wine and whiskey studies.
Thank you so much.
Okay, thank you, Dan. I've got a question in the chat from V. And the question is, can you use a mass spectrometer to decipher molecules that make up scent?
Well, it depends what you mean by makeup. You can of course measure the composition of something, like what molecules and the amount of there is in there, and of course the volatility of the compounds. But that will not necessarily give you a structure in a model. So maybe can you rephrase the question so that I know what you mean by what you get out of the.
So we'll see if that comes out, because that was in the
Okay.
chat. So maybe there will be a follow-up we can see on that. Why don't we turn now to Sam Fletcher? Sam, you should be able to talk now.
Hi, hello. I had a question about one of the arguments that you made at the very end, the Laplace's demon argument. So one of the things that I took away from your talk, which I think I learned more from than any other talk in recent memory, so thank you so much for that.
Thank you.
Is that one of the reasons why these machine learning models have been spectacular failures is that there are so many more features and interactions, particularly regarding the biology of olfaction, that are just not even in their data sets. And so there's no way that they would have been able to do anything with them, particularly because they're, as you've argued persuasively, they're so important for understanding olfactory perception. It seemed, though, that you wanted to then make an argument that even if we had all of the relevant correlations with the details about the different types of olfactory sensors, the different way that compositions and ratios of different molecules influence that, that even in those cases, we would never be able to, using machine learning techniques, predict how people would perceive smells. But I didn't quite see I didn't quite see why that was so. So I was hoping you could say more about that.
Oh, thank you very much for the chance to clarify that, because yes, there are actually several reasons. One thing is not just simply because of the point was really to highlight that without the biology, the chemical data will not give you an answer. But when we talk about the biology, it's not just simply that we would have to know all, like, the entire genetic makeup of the olfactory system, but we would also need to know the combinatorial coding, which means you don't just simply have a molecule with a smell, but it's encoded by different receptors. Now, these receptors can have different sensitivity towards that molecule, which link to the genetic diversity. So you might have at some point even mutations in the human olfactory genome, which it's one of the most highly, when it comes to genetic diversification, one of the most highly diverse systems. So this is what I mean by Laplace demon. You would have to have some kind of overarching perspective of the past, the present, the future of all possible genetic combinations you might have in this system. Again, you have 400 different types of receptors with different sensitivities potentially involved. This is unfeasible to get with machine learning without that kind of. Where would you get that data set from biology? Where would you get it from? Even let's say in let's say 100 years, you've got a mutation near one of the receptor genes. You can't predict that via machine learning, but that mutation might change, let's say, how you start perceiving vanilla. Maybe you get a mutation that at some point leads the receptor to say, oh, this is not vanilla, this is horse urine or something. So there's an unpredictability involved that cannot be covered by just covering the chemical complexity with machine learning. And even just having a limited amount of biological data, and at the moment it is limited, would not help it. And even having some potential, well, we might just need more data, would not suffice to get at the causal issue, namely the sensitivity that's informed by the genetic and genetic diversity, and how really the same odorant can have different qualitative or can lead to different qualitative perceptions based on the receptor behavior.
I see. So even if we focus, so what if we just focused on a particular we abandoned the project of how all possible odor perceiving systems would work, which I take it is maybe like a stronger, bigger goal than maybe the machine learning folks have in mind, which is rather how do like relevant populations with sufficiently low, levels of mutation, with some fixed level, right, of I'm not expressing myself very well here, but
it sounds a little bit like a physicist who says, imagine that's a cow drawing a circle on the blackboard. The problem is, well, you can build an artificial model that could be olfaction, but it's not olfaction. The question is, why would you do that? I mean, yes, you can do it, but
thanks.
Sorry, Ava was a bit too abrupt, but I mean, what you were saying is kind of something I got quite a lot of pushback from when I gave an earlier version of this talk to the informatics colloquium where at IU, where I thought, well, if I'm bold, I'm talking to the people who do machine learning to see what they think. And I got a very good colleague and friend out of that who's a bioinformatician who first went like, I'm not so sure. And they was like, okay, fair enough, good point. But one of the people also had a similar point with you. What if we just reduce the data set? What if we do that? The point is, it's not a matter of how much data you have. It's the causality behind connecting the different data sets that is essentially contingent on the behavior of the receptors. Everything else is, I mean, it's the old saying, correlation, causation, so to speak. You get a sufficiently enough correlation, fair enough, you might get some interesting correlations out of that, but it would be wrong to say that this is a way to decode how olfaction works. You might get some kind of leads, some hypothetical leads to what something could smell like, because you've got a sufficient overlap in structure-odor relations between people, but that is not an explanation, or it doesn't lead to structure-odor relations in the causal sense.
Yeah, that makes a lot of sense, although I guess I would have thought that most of the machine learning folks are interested in prediction rather than causal structures. But if one is interested in causal discovery, there's lots of interesting algorithms that are being, developed to try to extract causal information from large data sets. So it's not totally hopeless, although there's a lot of...
Well, the problem is that the prediction is, of course, what you're trying to... Well, you can't have a prediction without having the causality. In this case, like the most successful study was the 2017 study, and their success rate was 0.3, which is not that great.
Okay, Sam, did you want to follow up with that further?
No, that's okay.
Okay, great. Thank you for
but thank you for your question.
Yeah, and what I want to do is I want to briefly highlight, there's another question in the chat that I think you just addressed, but I wanted to double check. It said, the way machine learning is used for modeling olfaction is not working. However, do you think there is any room for machine learning in this topic applied in a better way, perhaps one that can account for biology in some sense? I take it this is related to some degree to what Sam just was talking about.
I'm not saying that machine learning is useless, so I should be more upfront about that. The problem is what you use it for. It's a tool that gives you hypothetical leads. The problem is that it's been mainly used in recent years as in a way I don't think is warranted. So I actually think that the recent trends towards employing machine learning in olfaction is actually great, because it might help us to figure out what part is complexity, what part is misunderstanding of causality. My issue with the current models is really that they're really modeling the wrong thing. So it's not against machine learning per se, but against how it's used and what we try to get out of that, if that clarifies it.
Great, thank you. Let's go next to Yoshi. Yoshi, you should be able to talk now.
Can you hear me? Okay, thank you. Yeah, thank you very much for the very interesting talk. I want to hear a bit more about the comment on case studies, one of the 3 points close to the end of your talk, partly because case studies based philosophy of science is my favorite kind of philosophy of science. So I'm interested in that point.
Oh, thank you, because this is something I started to worry and think about more and more. And I should maybe qualify this kind of drop at the end. It has to do with the fact that over the last years, I often heard these comments about my work in olfaction. Well, it's just a case study, and how do you extrapolate it to other case studies? And I noticed there's a different use of empirically informed philosophy of science in the community. Some people use a case study to make a point about a broader philosophical study or argument or concept, while there's also another way of doing something, and it's maybe not a good way to call this a case study. So I've been working on olfaction for a decade, a bit more than a decade now, where for me it's not a case study for a philosophical argument, but more to use philosophical thinking in the field itself. And there are more areas where this is actually would benefit the scientist, it would help the scientist, it would also help philosophers to rethink what philosophical methodology and philosophical work is and can do. And that brings me to a broader question, like how do we think about the role of a case study as a case study, or a case study as a way to do philosophy in science? And what larger consequences we might be able to derive from that for what philosophy of science is and can do, and even to what extent we might be able to do something like a natural philosophy for the 21st century, which is more than just having case studies to make a point, but really, can we change the way we do philosophy even in a modern way? And this is kind of more of a, what does that mean for the kinds of expertise we need to acquire? Because a lot of these things cannot be done just by doing philosophy and applying philosophical arguments. And I used to a little bit harshly say that when I entered the lab of Stuart, within the first week, I noticed how much my classical training in philosophy of science failed me. It failed me to understand what was going on. And there was a lot of struggle back and forth to communicate between the scientists, the philosophers, what's going on, such that it was not just simply the way we handle case studies in philosophy, there was something else going on. And we see a similar problem arising with people now wondering what's going on with COVID, where people see like, well, there's constantly some kind of a new study coming out with science communication, people are confused. And you had Jacob Stegenga recently in the BGPS blog saying, well, is this kind of an issue of a new science, a fast science? And I'm like, no, this is not new. Anybody who has worked on science at the frontiers where the question's unresolved knows that this is the case. This is what happens in the kitchen. And now the public has been treated to what happens in the kitchen and not just the finished dish. So we just didn't get exposed to, oh, we have a vaccine. We saw all the kind of, we have no idea how this virus is working. It might work like this, it might work like that. So the case study is mainly about how we link empirical philosophy and philosophy of science to the areas in which we think we might be able to apply it, and how we also need to differentiate between case study for philosophy and maybe philosophy contributing to an ongoing field, if that makes sense.
Yeah, thank you very much. That's very interesting.
Okay, we have a couple more questions that I'd like to squeeze in before we finish. The first is a clarification of the earlier question about can you use a mass spectrometer to decipher molecules that make up scent? V says, I meant the structure of molecules inside of something such as tannins inside of fruits. If you had perhaps urine, could you break down the biology of a human and determine the type of scent that they would be receptive to?
No.
Okay, we can make that be the quick answer, I guess, at this point.
It happens. I mean, as you can see, I've got either very long answers or very short ones, but that's the shortest I can do.
I think that's a good thing because we need to keep moving. Let's go to a question from Susan Sterrett. She says, what relevance does the literature on memory and the sense of smell have to your work?
If somebody would crack the connection between smell and memory, that's the next Nobel Prize. Actually, this is not this particular talk today, but it features quite a lot in the book when it comes to looking at smell as tags for experience, because we are not just simply memorizing perceptual objects or qualities, but situations. When you have a really good glass of wine, for instance, and later you see that wine again, like, oh yeah, I had this before. You don't really remember the quality. You don't go, oh yeah, there was a nice raisin and well, a nice vanilla or oak note. You remember where and when you had that wine, the people, the atmosphere. Sometimes you even remember the lighting. So a lot of odors that we memorize are often as tags for experience. So it features in my work, but not necessarily in this part of the talk. But there is stuff A, more hopefully coming out, and B, already partly in the book.
All right, we've got 2 last questions. I will ask them to be as quick as possible so we can squeeze them in before finishing. Bill, you are next, and you should be able to talk now. Bill, can you hear me? You can unmute and talk now.
I got it. Ann-Sophie, that was phenomenal. I spent my career studying complexity, and you've exceeded my threshold. In any case,
Thank you.
let me say, all right, and this connects with memory, but how is sensory generalization possible? That is, I recognize the smell of apples. I can smell when there's a bad apple. I can tell when my cider is getting a little vinegary rather than spiked. I can, distinguish cheeses and all those. And I can tell, yes, this is actually the same Roquefort I had last time. I like it. So how is sensory generalization on the one hand and discrimination for fine modulations possible?
I emphasize the difference. That's a very good question. I emphasize more of the differences in terms of variation. But one reason why we are, say, able to say, oh, that's an apple, why you and I will go like, that's an apple, is the combinatorial coding. There is sufficient overlap of receptors by which we detect something. So there's a nice twist in terms of we're able to detect the same things because we've got a sufficient overlap in terms of receptors and receptor expression, but also enough variation so that your apple literally doesn't smell like my apple. So it's a nice empirical answer to the qualia problem, so to speak.
Yeah, no, I'm not supposing that we have the same apple smell. Yeah.
But as you say, like partly it's because we have a sufficient overlap of receptors, and partly because we also a lot of the ways in which by which we characterize and learn sensory qualities in olfaction is by learning by association. And so this is why we grow up in a similar enough community with the same kind of food smells, the same kind of flower smells, etc. So there's also a certain form of cultural and conventional element involved in that. It's biology and culture. I never separate these two because there's a lot of overlap when it comes to the associative learning as well as the biological overlap of receptors. So this is why you can detect the same things over time. Sometimes it's harder for you to identify it in terms of name it, but you will go like, it's familiar. I've known this before. I've smelled this before,
yeah.
and so there is enough overlap. By the way, I have to soon actually also run off because I'm getting my first vaccine.
Okay, so thank you, Bill. And we'll go now to Charlie for one last quick question. Charlie? Go quickly, Charlie.
Really quick.
Very
This garbage in, garbage out, in my humble opinion, applies to the vast majority of the AI literature. That's terrible literature. It's totally irreproducible. It's just claims. You don't know what they did. You can't find out what they did. You don't know whether they were cherry picking. Almost certainly there was a lot of that. They are the only area that hasn't even heard of the reproducibility crisis and couldn't care less. They've got a long way to go.
I was too polite to say that, so I just let you say it and grin and go like, yes.
Yeah.
Okay, thank you, Charlie, for that last point. And Anne, I want to thank you for joining us today. A really wonderful talk. Everyone appreciates the time you invested and the way you answered the questions. We hope the best as you go off to get your vaccine. And for those who are on the line, please do join us next time for our next presentation, which you can find out about on our website for the Minnesota Center for Philosophy of Science. Take care, everyone, and have a good weekend. Bye-bye.
Thank you very, very, very much. Have a lovely day.